In essence, it is a gene therapy that is activated by the increase of brain activities, for example, as you would see at the seizure initiation stage.
Today we have neuroscientist and epilepsy researcher Yichen Qiu of the Legnani lab at UCL explain to us her research into stopping epileptic seizures in space and time through gene therapy.
Hi, thank you for having me. My name is Yichen Qiu, and I am working as a postdoc research person at the Institute of Neurology at Queen Square in London. I'm with the Department of Clinical and Experimental Epilepsy. So we research into the mechanisms of the disease, the epilepsy disease, and we are looking into new and innovative therapeutic strategies for treating epilepsy.
Fabulous. You say we. How big's the team that you're on?
So as the title suggests, we are kind of split into two sides. So half of the department is working on the clinical side and half of the department is working on the experimental side. So it is really close relationship between the clinical and research. I would probably say it's about like 30 people on our experimental side. I think the clinical side is a little bit bigger. So a lot of people.
Yeah, but you are sorely needed, so it makes complete sense. And so today we're going to talk specifically about, you were recommended to me with your paper called On-Demand Cell Autonomous Gene Therapy for Brain Circuit Disorders. Tell us about that. What does that mean to the world and to other clinicians and people affected by epilepsy? What have you discovered?
Yeah, the title is including a lot of different components, and we tried to put everything into one row. But in essence, I'm generally speaking, we are kind of researching into gene therapy for epilepsy. So in terms of gene therapy strategies, we generally would, when we design gene therapy strategies, we would mainly consider two components. One is how we're going to get in there, how are we going to get to the point where the seizure is or where the epileptic focus is, and another component will be what are we going to put there, like what is the cargo. So in essence, it is a gene therapy that is activated by the increase of brain activities, for example, as you would see at the seizure initiation stage. And as a result of it, it then expresses a protein which accounts for an ion channel. And the expression of that ion channel is going to calm neurons, calm the neuronal circuits. And as a result of it, hopefully it will stop the seizures before it spreads to the rest of the brain.
How did the planning of the study happen? How were things laid out? How long did things take? At what stages did you make any discoveries?
One of the sort of the driving force behind us trying to come up with new or innovative gene therapies with the fact that there is already conventional strategies which will be taking the pills, so the medications, that has been pretty effective. So the reason for that is that only about 70% of the patients respond to drugs. So that means the remaining about 30% of the patients, they don't really respond very well to medications or the combinations of medications. So for those patients, they don't really have many options which is available for them. So that's why we think that there is a huge need for new therapy strategies like this. So I guess I'll probably kind of make a metaphor. So bear with me. So in a way, or like how I see how this gene therapy works is, I guess I imagine like a brain is like a hotel, like a lot sure it's a hotel, with hundreds of thousands of rooms, which are like neuron cells. And as we know that neuron cells are connected together, and same as hotel rooms, they are connected by corridors, complicated structures, staircase, and everything like that. And when there's like a seizure happening, it's almost like a couple of rooms are on fire. So because it's a luxury hotel, everything's laid with carpet, it spreads really, really quickly from one part of the room to other parts of the room. In some cases, you might have multiple sites where the fires start together. This is when, if you think about conventional medications, that's when you call 911, and you have the firefighters coming, and they pour so much water all over the place that it puts out the fire. However, even though the fire is put out, the problem is then the hotel needs to be restored. Like everything is good anymore. So in our case, we're kind of doing it in a way that instead of waiting till the time where things have gone so bad that you have to call the firefighters, we kind of just installed the sprinklers in every room. So it's not doing anything when there's no fire. However, when there is a fire, what it does is that it detects the smoke or the fire, and it will turn itself on, and it tries to put out the fire before it spreads to the rest of the hotel. So in that way, hopefully you don't get the fire at the same time. You still keep the hotel functioning.
And so you mean, like, for instance, so with, many regular sort of drugs, that anti-seizure medications that people take, it feels like they're dulling down the whole ecosystem of one's brain consistently just to try and avoid seizures. So that's kind of like the water, the foam coming from these fire extinguishers, right? So, but the treatment that you're proposing or that you studied, it fires the foam or the water at a specific time to avoid seizures. Is that what you're saying?
Yeah, so it's specific both in space and time. In space, meaning that it is hopefully not going to be activated in every cell. Specific in time means that it is not always on.
So it zaps the dodgy bit that is causing the seizures, and rather than the whole shebang being affected as it is with regular medications.
Yeah.
So you're talking about in this paper, and I'm bringing it up literally now, so we're talking about the KCNA1 gene in particular in this study. So tell us about this particular gene and why this is so impactful when it comes to certain epilepsies, and tell us which epilepsies it has an impact on.
In this case that we are using, or we are looking into temporal lobe epilepsy. So in this case that the target that we're choosing being the KCNA1, is for the reason that KCNA1 is a gene that codes for Kv1.1 potassium channel. So this channel has a pretty nice role in terms of calming down the neurons when it is expressed. So in this case, that the neurons with this channel expressed is going to be less active or less likely to become super duper duper abnormally active. And in that case, we hopefully would reduce the possibility of that cell being involved in the seizure. And on a populational scale, we hopefully would increase the threshold for seizure in that case.
Tell us of this precise outcome, if you can, in a few words, of this study, and what would be the next steps potentially?
In our paper, we first look at the gene therapy strategies in vitro models, so on neuronal cultures, which we saw that there's a general decrease in neuronal excitability in the cellular population. And then we moved on to preclinical experimental model, which we saw that there is a significant reduction in seizure frequencies. So that has kind of given us a lot of hope that this is doing something interesting to the disease. And so in addition to that, we also did something which is quite interesting. We applied this gene therapy strategy to organoids. So organoids are the really nice and sweet and kind of, I think people sometimes will call it brain in the dish.
Yeah, think of the Petri dish, right?
Yeah, that'll be the hope. But in general, it is a 3D structure derived from human-based cells, with the hope that it will more likely to create a environment or condition more similar to sort of human brain. So we actually saw there is a reduction in the apoplectiform activities in the organoid experiment. So that was also really exciting results. So I think in terms of the next steps, obviously we'll have to look closely in terms of the to really understand. In addition to it being effective, obviously there's the safety side of it, like to understand the toxicity of it and to really do a further kind of analysis into what is it doing and what is the mechanism of it and why, also to answer the questions of like, why is it so effective? Is it better than other things, or is it something even better than this? So there's kind of like loads of branches that we could potentially.
You talk about potentially branching into these areas. Will that be happening, or is the study at the moment, as far as we know, kind of at an end, or what will you be doing?
The hope is that we will be carry on studying it and carry on understanding how it works, and with the hope that eventually, with the aim that maybe in the future this could be something useful for the clinical side.
Yeah, I think what's worth everybody remembering, keeping in mind, is that although these are wonderful, exciting, like just great results, at the moment we don't know of the potential long-term impact. We don't know of any side effects. We don't know if it would necessarily be great in humans, or if it's going to be, because with the epilepsy, seizures aren't everything, with the impact on mental health be awful, or we just don't know, right? And that's why research takes a while.
Yes, I mean that's why we're still kind of putting a lot of effort into understanding all of those things, and trying to see if this is something that can be carried on to the next stage.
Thank you so much to Yichen for sharing with us and explaining her cool research into gene therapy for temporal lobe epilepsy. Let's all stay tuned to hear how things progress. If you'd like to learn more about gene therapy and indeed the Legnani lab where Yichen works, do check out our episode on gene therapy with the man himself, Gabriele Legnani, Principal Research Fellow and Professor of Translational Neuroscience. We will put the links in the text.